Hello everyone and welcome to my YouTube channel Perfect Lab. In today's video we will learn about the differential media as a type of culture media in microbiology. We will also look at the composition of this media and some common examples used in the culturing of microorganisms. So let's get started. Let's start our introduction by looking at what a differential medium is. So a differential medium is a culture medium that allows the growth of different microorganisms and enable them to be distinguished from one another based on observable differences in their colony appearance or by chemical reactions. Now let's look at the component or the composition of a differential media. So the first component happens to be basal nutrients, and these basically provide the essential nutrients for microbial growth. And examples are peptone, which supplies amino acid, peptides, nitrogen, and energy needed for bacterial growth. We also have beef extract, which also provides vitamins, minerals, and organic compounds to promote bacterial growth and metabolism. And we have yeast extract, which is rich in vitamin B, amino acid, and supply growth factors to support most fastidious bacteria to grow. The second component are differential agents, and these are basically substances that produce visible differences between organisms when cultured on this medium. An example are dyes such as eosin methylene blue that directly stain cell walls, and pH indicators such as neutral red and phenol red, which change color as a result of metabolic byproduct. We also have esculin, which is used as a differential substrate to identify bacteria that produce the enzyme esculinase, and urea to identify bacteria that also produce the enzyme urease. And finally, we also have other components that support the medium function and the medium consistency. An example, we have sodium chloride, which basically maintains osmotic balance. We also have agar as a solidifying agent, and in some cases, selective agents that will select organisms and inhibit the growth of some other organisms. So let's look at some examples of differential media. So the first differential media we will look at is the blood agar. And in the blood agar, the differential agent in the medium is the red blood cell. And these red blood cells help to differentiate bacteria based on their hemolysis pattern. Example, so for bacteria that undergo beta or complete hemolysis, show a clear zone on the agar medium, and those that undergo alpha or partial hemolysis show a greenish zone on the agar medium, and those bacteria that undergo gamma hemolysis or are not able to hemolyze red blood cells form red colonies on the agar medium as shown here. So the second differential medium we will look at is the eosin methylene blue. And in the eosin methylene blue agar, the differential agents in this medium are lactose, eosin Y, and methylene blue. And these differential agents help to differentiate bacteria based on the degree to which they ferment lactose. For example, so bacteria that are strong fermenters of lactose show a green metallic sheen on the agar medium as shown over here, whereas those that are weak or non-fermenters also show or appear pink to colorless on the agar medium. The third differential medium we will look at is the mannitol salt agar, and the differential agents in this medium are mannitol and phenol red. And these agents also help to differentiate bacteria that ferment mannitol to produce acid and those that do not ferment mannitol. So for bacteria that ferment monitor turns the medium yellow as shown over here as a result of the production of acid, and those that do not ferment monitor remains pink or red on the agar medium. So the fourth differential medium we are looking at is the MacConkey Agar medium, and the differential agents in this medium are lactose and neutral red, which help to differentiate lactose fermenters bacteria from non-lactose fermenters. The lactose fermenters produce pink to red colonies on the agar medium, whereas the non-lactose fermenters produce or form colorless to pale colonies on the agar medium as shown over here. The next is the SLD agar medium, and in the SLD agar medium, xylose, lysine, and phenol red are the differential agents, and these agents also help to differentiate enteric pathogens based on their sugar fermentation and the production of hydrogen sulfide. Example, so bacteria that ferment xylose, such as Salmonella, form yellow colonies on the agar medium after incubation, and those that do not ferment xylose form red colonies on the agar medium, and the production of hydrogen sulfide is usually seen as black centers or spots on the agar medium as shown here. So the next or the sixth differential medium is triple sugar ion, or in short TSI agar medium, and the differential agents in this medium are glucose, lactose, sucrose, phenol red, sodium thiosulfate, and ferrous sulfate. And these differential agents basically help to differentiate bacteria based on carbohydrate fermentation, gas production, and hydrogen sulfide production. And the last and seventh differential medium we are looking at is the biochemical agar. So in the biochemical agar, the differential agents here are esculin and ferric citrate. And these differential agents help to differentiate bacteria that have the ability to hydrolyze esculin from those that are not. And the formation of black or brown-black colonies on the agar medium indicates esculin hydrolysis, whereas no color change in the agar medium shows negative result. So the key point we have to know in differential media is that they contain specific agents that produce visible differences in color, in colony color, zone of clearing, and other reactions based on microbial characteristics. And this media really matters in microbiology because they help microbiologists to distinguish between different organisms based on their metabolic and biochemical activities. So thanks for watching my video, and if you found this video helpful, please like, share, and subscribe to Perfect Lab. Leave your comments or questions below, and don't forget to scan the code on your screen to follow us on WhatsApp for more laboratory updates and tutorials. See you in the next video.